Hey class, it's another beautiful week and we are going to move on to the end of our digestive system lecture series and talk about metabolism and energetics this week. Metabolism is the sum of all chemical reactions that occur throughout your body. So throughout the entire organism is your metabolism. Cellular metabolism are the chemical reactions that take place within individual cells, and those provide the energy needed to maintain homeostasis and essential functioning of each individual cell. Your overall metabolism helps to maintain homeostasis throughout your entire body. So we have cellular metabolism and then overall general metabolism. A couple of vocabulary words: catabolism, which you'll see here. Oh, where's my guy? Here. That is the breakdown of organic substrates. So that's breaking down. So if you're eating carbohydrates, carbohydrates are going to get broken down into simple sugars. Fats are going to get broken down into fatty acids and glycerol. Proteins are going to get broken down into amino acids. So those are all catabolic reactions where you are taking something large and breaking it down to something small. And then anabolism, which here is the synthesis of new organic molecules. So taking amino acids and putting them together and synthesizing new proteins. Taking monosaccharides, individual small sugars, putting them together and making disaccharides and trisaccharides and other carbohydrates. I always remember this when I think of bodybuilders and they take anabolic steroids to get bigger. So if you can think of an anabolic steroid, anabolism makes things bigger, so you're making new molecules. During cellular metabolism, our cells are always breaking down their individual parts and replacing them. The only exception is DNA. Cells do not break down their DNA. The DNA remains intact, but the rest of the cellular parts are getting broken down, recycled, and renewed, building new ones all the time. We have a nutrient pool in our body. So we have one in our cell, a cellular reserve, and then we have other nutrients throughout the body that we can call upon when we need them. So these are basically reserves of organic building blocks that you can pull out of reserve in order to help build proteins or renew your plasma membrane, all of those sorts of things. We do have reserves within our body to manage that. So we are not running, nutrient for nutrient as we eat. Here's a little diagram to show us that. So here we have organic molecules, so amino acids, lipids, and simple sugars that are coming into the body. So these are out in the interstitial fluid. They enter the cell, and we have the nutrient pool. So that's all of our extra. So as we build things, we have anabolic reactions. We are utilizing the nutrients in the nutrient pool to maintain and repair, for growth, for secretions, and for reserves, like our fatty reserves. And then during catabolic reactions, we are breaking down all of these reserves and adding them back into the nutrient pool or utilizing them within the cell for whatever we need. Catabolic reactions generally occur in the mitochondria, and the breakdown of organic molecules results in the formation of ATP and the release of heat. So we're going to talk a lot today about how ATP is created. We need a lot of ATP in our body for locomotion, contraction, endocytosis, exocytosis, everything that happens in our body, not everything, but most of the big important stuff that happens in our body is powered by ATP. So the breakdown of nutrients within the mitochondria generates our ATP for us. This one is showing us we have our nutrient pool. So in our nutrient pool, we have fatty acids, we have sugars in the form of glucose, we have amino acids, and those molecules can be pulled out of the nutrient pool in order to create triglycerides, glycogen, and proteins, or they can be pulled out of their triglycerides. They can be broken down. Glycogen can break down to glucose. Proteins can break down to amino acid. All of those things can be broken down further into small carbon chains. Those small carbon chains can then enter the mitochondria, go through what's called the citric acid cycle or the Krebs cycle, and then be sent to the electron transport chain where ATP is created in mass. Like I said, most of our ATP is generated within the mitochondria of the cell. The cell feeds the mitochondria with items from the nutrient pool. It's a very picky eater though, so it needs very specific and small organic molecules that it can then process and create ATP from. So we're going to look at what different molecules can go through the Krebs cycle and therefore produce ATP. So this process is going to begin in the cytoplasm. We're going to be looking at breaking down molecules. So we need them to be broken down small enough to get into the mitochondria. So chemical reactions within the cytoplasm are going to take the nutrients from the nutrient pool, break them down into smaller fragments so that those small fragments can then enter the mitochondria and be broken down. The mitochondria are going to break fragments down further, and within that breaking down, they are going to generate ATP, but as a byproduct, they're also going to generate carbon dioxide and water. In order to generate ATP, we need to go through a couple of different processes. The first one is the citric acid cycle or the Krebs cycle. And the second one is the electron transport chain. And so this is a process of first you go through the Krebs cycle, and then the Krebs cycle is going to send byproducts to the electron transport chain, and the movement through the electron transport chain is going to end up creating ATP in the end. So we're going to start with carbohydrate metabolism. This generates ATP and other high energy compounds by breaking down carbohydrates. So generally we say glucose, which is sugar, plus oxygen can get broken down and yield carbon dioxide and water. This happens in many small steps, and each step is going to release a small amount of energy, and that energy is going to convert ADP, adenosine diphosphate, to ATP, adenosine triphosphate. One molecule of glucose can net 32 molecules of ATP. This, I think, I'm going to go with 32. That's what your book says, even though the PowerPoint says 36. So please cross out 36 and write in 32. The first part of the glucose breakdown is glycolysis. And then from glycolysis, we go through the citric acid cycle. And then from the citric acid cycle, we progress through the electron transport chain. And that's how we end up with our 32 molecules of ATP. So we're going to break down each one of those steps and show you what's happening at each level. So glycolysis is first. It breaks down glucose in the cytosol. So glycolysis happens in the cytosol. The rest of this happens in the mitochondria. So in the cytosol, glycolysis breaks down glucose into smaller molecules that can be used by the mitochondria. And this does not require oxygen. So this is the only step that does not require oxygen. The rest of it, the citric acid cycle and the electron transport chain, do require the presence of oxygen. But glycolysis is anaerobic. It breaks down a 6-carbon glucose into 2 3-carbon pyruvic acid molecules. And then the pyruvic acid is further broken down to pyruvate, and pyruvate can then be broken down further and enter the citric acid cycle. So I think that I reminded you previously that this is a challenging week. So I hope you got your focus on today. So first thing we started with is glycolysis, which is the breakdown of glucose in the cytosol, does not require oxygen, takes 1 6-carbon glucose and takes it into 2 3-carbon pyruvic acid molecules. So here's our steps in glycolysis. So this is all taking place in the cytosol or the cytoplasm. And we don't need to, all of these chemical forms here, I would not ask you to know those. So if you are into chemistry and you want to know all this, then by all means knock yourself out. But we can just focus on well, what's happening at each step. So step one is the breakdown of glucose or the addition of a phosphate. So the addition of a phosphate is called phosphorylation. So the phosphorylation of glucose adds a phosphate group to the glucose molecule, and that requires 1 ATP. So the glycolysis actually requires 2 ATP to complete this whole pathway. So the first step is phosphorylating the glucose into glucose 6-phosphate, and that utilizes 1 ATP. The second step is another phosphorylation. So we're adding a second phosphate. So here's our first phosphate, and now we're adding our second phosphate, and we're utilizing a second ATP. So right now we are down 2 ATP. Then what happens is the 6-carbon chain, so those C's there are 6 of them, those are carbon molecules, gets broken down in this third step into 2 3-carbon chains here. Then we phosphorylate again, so we have 2 more phosphates. 1 is added to each of those chains, so now you see we have a phosphate on either side of those chains. And in that step, we have 2 NAD. NAD is a coenzyme, and the NAD is picking up a hydrogen molecule and becoming an NADH. And when NAD, the coenzyme, holds an additional hydrogen molecule, it makes the NAD hold energy. So now this NADH has the ability to go enter the electron transport chain and generate ATP. So what we're going to be looking for in all of these steps through glycolysis, the citric acid cycle, and then the electron transport chain is the formation of ATP, but also the formation of NADH, and then later the formation of FADH2. Those are 2 coenzymes that have the ability to hold additional hydrogen, and that gives potential energy to those coenzymes. So our fourth step here, we phosphorylated our 2 3-carbon chains, and in the process, we created 2 NADH. Step 5, we are going to break one of those phosphate bonds, so dephosphorylate, and that is going to create 2 ATP. So up here on the top, let's see. Up on the top here, we were minus 2 ATP. Wow, this is really hard. And here we are plus 2. So really at this point, our net ATP is 0. Hopefully that makes sense. So we utilize 2 ATP up top in order to phosphorylate our 6-carbon chain. And then we used an N2 NAD to phosphorylate our 3-carbon chains. And in that process, we created 2 NADH here. And then dephosphorylating our 2 3-carbon chains is going to produce an additional 2 ATP. So right now we've used 2, we've made 2, so we have no net change in ATP. So now these 2 molecules in step 6 are going to get rearranged, and so instead of the phosphate being over here, the phosphate's going to be on top, and that rearrangement allows for the release of 2 water molecules. So water is a byproduct of glycolysis. And then the last step is processing of that molecule, releasing those phosphate groups, and in the process we are creating an additional 2 ATP, and our end product here are 2 pyruvic acid. Let me erase that so you can see our pyruvic acid is what we're looking for. So glycolysis is taking a glucose maybe I'll do that in a different color, taking a glucose molecule and then breaking it down to 2 pyruvic acid molecules. And in the process, it's going to create 2 ATP, and it's going to create 2 NADH. So our summary down here, steps 1 and 2 utilize 2 ATP. Step 5 creates 2 ATP. Step 7 creates 2 ATP. So our net gain is 2 ATP out of glycolysis. And then we also have those 2 NADH. So everything we just talked about with glycolysis was step 1 in our breakdown of carbohydrates or breakdown of glucose. Step 2 is ATP production within the mitochondria. So in the mitochondria, we require oxygen in order to absorb and break down pyruvic acid molecules. So remember, the pyruvic acid was created at the end of glycolysis, and then the pyruvic acid absorbs into the mitochondria and is then broken down further. So the hydrogen atoms of the pyruvic acid are removed by coenzymes, and the hydrogen atoms become a primary source of energy gain once we get to the electron transport chain. Carbon and oxygen atoms are also removed and released as carbon dioxide in the process of decarboxylation. So decarboxylation is removing a carbon dioxide. A couple of other terms that I want to just bring up at this point. Oxidation is the loss of hydrogen or the gain of oxygen. When you lose hydrogen or gain oxygen, you decrease your potential energy. And then reduction is a gain of hydrogen ions, which increases your potential energy. So we have these reactions that are called oxidation reduction reactions or redox reactions. And basically what they are the transfer of hydrogen ions and electrons between 2 molecules. So when you take a hydrogen ion from one molecule and you transfer it to another molecule, the first molecule that has lost that hydrogen ion becomes oxidized, and the second one, which has gained the hydrogen ion, becomes reduced. And a reduced molecule has more potential energy than an oxidized molecule. A way to remember that is with the mnemonic OIL rig. OIL, O-I-L, stands for oxidation is loss. So we're talking about hydrogen ions. So when you oxidize, you lose a hydrogen. And reg is reduction is gain. So when you are reduced, you gain a hydrogen ion. So that's all in your book, but I want to make sure that you guys do take a look at that. So within the mitochondria, there's a double membrane surrounding the mitochondria. The outer membrane has large pores in it, and they are permeable to ions and small organic molecules, such as pyruvic acid. And then the inner membrane is lined with these carrier proteins. And the carrier proteins are move the pyruvic acid through the membrane into the matrix of the mitochondria. The space between the outer membrane and the inner membrane is called the intermembrane space. The function of the citric acid cycle is to remove hydrogen ions from organic molecules and then transfer them to coenzymes. I would read that 3 or 4 times. You might see that again. The function of the citric acid cycle is to remove hydrogen ions or hydrogen atoms from organic molecules and transfer them to coenzymes. During that process, 2 carbon atoms of the acetyl group are removed or released via molecules of carbon dioxide, ending with a 4-carbon molecule, which is ready to receive another acetyl group and form acetyl-CoA. So the main thing you want to come out of here is acetyl-CoA is what we need to go into the citric acid cycle. So when we break down glucose into two molecules of pyruvic acid, the pyruvic acid is decarboxylated, that loss of two carbon atoms in carbon dioxide, so that's decarboxylation, and then it can be converted into acetyl-CoA. And the acetyl-CoA is what enters the citric acid cycle. During this breakdown, a single molecule of ATP is produced each time an acetyl group is processed by the citric acid cycle. So here's our pretty picture. We have the pyruvic acid that was the end product of glycolysis, and through coenzyme A, and coenzymes and enzymes in general are what allow for reactions to happen to change one molecule to another molecule. So coenzyme A interacts with pyruvic acid. We have a decarboxylation, so a loss of carbon dioxide, and that is going to create acetyl-CoA, and it also is going to create one NADH molecule. Then the acetyl-CoA enters the citric acid cycle. It releases its coenzyme A and is a 6-carbon citric acid. That's the first molecule that gets formed, is our 6-carbon citric acid. That gets decarboxylated and releases 2 hydrogen ions, which then go and form 2 NADH. Now we're down to a 5-carbon molecule. That gets decarboxylated, releases 2 hydrogen ions, which then form an additional NADH. Then we release an ATP, and it's really what we're releasing is a GTP, which is guanosine triphosphate, I believe, and the GTP can easily be converted to ATP. So this is the one ATP that gets produced in the citric acid cycle. So now we're down to a 4-carbon molecule, and some additional processing goes on and releases 4 additional hydrogen. 2 of them become FADH2, and 2 of them go and generate NADH. So the entire citric acid cycle per pyruvate creates, let's see, let me just delete all this so you can follow along with me. All right, so each cycle creates 3 carbon dioxide, 1, 2, 3, creates 3 water molecules, which we don't see on here, but it does happen, creates 4 NADH, 1, 2, 3, 4, creates 1 FADH2, and then 1 GTP, which converts to an ATP. So that's the whole purpose of the citric acid cycle, is to create this 1 ATP and then these 4 NADH and this FADH2. And you remember that FAD and NAD are coenzymes. They can bind hydrogen, and then they bring those hydrogen to the electron transport system. If you're a chemistry buff and you like to see the actual molecular structure of all of these things, then this is the slide for you. If you are not, I would pretend you never saw this slide and just keep on going. So now we've liberated those hydrogen ions from the pyruvic acid in the Krebs cycle. So they need to go through a process called oxidative phosphorylation. Oxidation is the transfer of electrons, the loss of electrons, which we talked about before. And phosphorylation is the attachment of a high energy phosphate group to an ADP to produce an ATP. So ADP is diphosphate, has 2 phosphates. ATP triphosphate has 3 phosphates. So when we add a phosphate group to the 2-phosphate molecule, we then have a 3-phosphate molecule, and that ATP is our high energy compound, and that's where we can release energy and power all of our systems. So in order to do that, we need coenzymes, we need oxygen, and then we can generate ATP. So this produces about 95% of the ATP used by our body is going through the electron transport chain. So we've talked about NAD and FAD. They are coenzymes. The purpose of them is to attach to a hydrogen that gets released from the citric acid cycle and then deliver that hydrogen to the electron transport system, or which is also called the electron transport chain. Each hydrogen atom consists of an electron and a hydrogen ion. Electrons carry our chemical energy. So the electron transport chain is going to carry those electrons. It happens through proteins that are along the inner mitochondrial membrane. And the coenzymes NAD and FADH2, or NADH and FADH2, are going to release the proton, so the hydrogen ion, into the matrix of the mitochondria and give their electron to the electron transport chain. The proteins in the electron transport chain are called cytochromes. Cytochromes are a protein that surrounds either a copper ion or an iron ion, so they have a metal ion within them, and they are embedded in the inner membrane of the mitochondria. And what happens is the first cytochrome is going to pass an electron to the second cytochrome, that'll pass it to the third cytochrome, and so on, so that the electrons move through each of these proteins that are bound to the inner membrane. Along the way, many of these steps are going to release enough energy to pump the hydrogen ions into the intermembrane space. We're going to see a picture, and it's going to make more sense when we see this picture, just so you know. So as these hydrogen ions are getting pumped from the inside of the mitochondria out into the intermembrane space, that creates a potential, a transmembrane potential. So you have these positively charged hydrogen ions being pumped out, and so it's making the outside of the mitochondria positive as compared to the inside of the inner membrane of the mitochondria. So in the last step, those hydrogen ions diffuse back into the matrix through membrane channels that are the enzyme ATP synthase. And as the hydrogen ions rush back in through a process called chemiosmosis, the positivity comes across into the negative cell, and that releases a ton of energy, and the energy that gets released by the downward movement of the positive charge and the minimization of the transmembrane potential is going to be enough to create additional ATP. I know that was a lot of words all in a row, so we're going to break it down further. So this picture is going to show you that we have NADH has a hydrogen ion, and it's going to give its hydrogen to the to CoQ, which is going to then send the electron to cytochrome B. So these are our proteins within the chain. So this line is the inner membrane of the mitochondria. I much prefer the next picture, but I'm going to try to work you through this picture first. So NAD enters at the first cytochrome. So this FMN here is the first cytochrome. So it gives its electron to the cytochrome, and it releases its hydrogen into the, if we say this is the inside of the mitochondria, it releases the hydrogen into the mitochondria matrix. Then the electron gets sent down the transport chain all the way to the end. FADH releases its hydrogen into the second protein in the electron transport chain. And then it sends its electrons all the way down through the rest of the chain. At the very end, we accept oxygen along with these hydrogen, and we have a byproduct of water. All right, so this picture I like much better, as it shows you where we're talking about. So up here, this is the outer membrane of the mitochondria. You can see it's a phospholipid bilayer, just like our normal cell membranes. And then this is the inner membrane, another phospholipid bilayer. So it's two membranes within the mitochondria. And up on the picture up here, you can see there's the outer membrane surrounding the whole organelle, and then this inner membrane, which is folded inside. Our NADH is inside the matrix. So down here, this is all matrix inside the mitochondria. So this is cytoplasm of the cell. So we're outside the mitochondria. This is the intermembrane space between the mitochondria, and then this is all matrix down here. So I'm going to just delete all that. So the NADH has come from the citric acid cycle. It's been produced, and it's bringing hydrogen ions to the electron transport chain. So what it does is it releases its hydrogen ion here, and it gives its electron to this first protein. And you see these are like integral membrane proteins that make up the electron transport chain. So the electron has been given to this protein, and the hydrogen ion has been released into the matrix of the inner mitochondria. And then FADH2 is going to come in, which would be here, and it's going to give an electron to this second, or actually here, this is FAD. So it's going to give its electron to the second protein and give release its hydrogen ions into the matrix as well. So now we have two electrons that are going through the electron transport chain. And every time an electron moves from one protein to the next protein, that potential energy that gets released drives these hydrogen ions out of the inner membrane of the inner matrix through the inner membrane into the intermembrane space. So you can see another here, we have hydrogen ions getting pumped out into the intermembrane space. And here, this is a channel protein, so the hydrogen ions are coming out. So we're making this huge positive charge up in that intermembrane space. And in contrast, or in comparison, we are negatively charged now inside the matrix. And we know from our study of the nervous system that when you have that big transmembrane potential and say we're doing like the neuromuscular junction, the binding of acetylcholine opens the sodium channels and the sodium rushes into the muscle cell, and that sodium entry is going to trigger the second action potential down the sarcolemma. Hopefully that's a nice review and everybody hasn't blocked that out completely. So in this case, it's the same thing. So we have all of these positively charged hydrogen ions out here, and they want to come back in because it's negatively charged inside here. So what happens is this ATP synthase, which is here, is the coenzyme that has a channel to allow for hydrogen ions to re-enter the cell. And that influx of hydrogen ions back into the inside of the matrix of the mitochondria is going to generate enough power to produce additional ATP. So I'm going to delete all of my chicken scratch and just go over that one more time. So we're in the inside of the mitochondria. We have the outer membrane up here, we have the inner membrane down here, and the intermembrane space in between. The electron transport chain is made up of membrane proteins within the inner membrane of the mitochondria, and the purpose of them is to move electrons along from the first protein through to the end. The NADH was made in the citric acid cycle by the breakdown of pyruvate, And what it does is it gives its electron to this first protein here, and then it dumps its hydrogen ions into the matrix of the mitochondria. Then the FAD is going to give its electron to the second protein, and it's going to dump its hydrogen into the matrix. So now we have hydrogen inside and nothing outside. So the transfer of electrons from one protein to the next protein generates enough energy to force this hydrogen out of the cell into the intermembrane space. And that happens multiple times along the way. So we end up pumping all of these positively charged hydrogen ions up into the intermembrane space. So we have a positive charge overall in the intermembrane space and a negative charge in the matrix of the mitochondria. This gives us a large transmembrane potential. And anything that has a concentration gradient or an electrochemical gradient wants to move down that gradient. So the ATP synthase, which is here, is going to open its channel and allow those positively charged hydrogen ions to rush back into the matrix down their electrochemical gradient, releasing energy and therefore converting ADP into ATP. This is an older picture from an older version of your textbook. So I'm going to point out right now that this is a typo. This 24 ATP here should say 20 ATP generated. So this 36 should actually say 32. So sometimes textbooks are wrong and they get corrected later, but we haven't quite updated the PowerPoint yet. So please change that 24 to a 20 and that 36 to a 32. So this is an overview of the entire system that breaks down glucose. So we start up top with glycolysis. Glycolysis is going to utilize 2 ATP and it's going to create 4 ATP for a net gain of 2 ATP. And glycolysis is breaking down glucose into 2 molecules of pyruvic acid. Next, the pyruvic acid is going to be broken down into acetyl-CoA, and in that process, it's going to create 2 NADH. We also created 2 NADH up at during glycolysis. Acetyl-CoA is then going to enter the citric acid cycle, where it's going to create 6 NADH, 2 FADH2, and 2 GTP. Because we had 2 molecules of pyruvate, so we're going through 2 cycles of the citric acid cycle. So therefore, we're generating 6 NADH, 2 FADH, and 2 GTP. Our NADH here is going to connect with our NADH here, and our NADH from glycolysis is going to come down. And so all of those are going to, along with the FADH, are going to enter the electron transport chain. Through the electron transport chain, the 2 NADH on top are going to generate 4 ATP. The 8 NAD from here are going to generate 20 ATP. And then the 2 FADH are going to generate 4 ATP. And then our 2 GTP are going to get converted to 2 ATP. So our net gain is going to be 32 ATP. So again, glycolysis first, citric acid cycle second, electron transport chain third. So we break down one single glucose molecule and we end up with 32 ATP at the end. An interesting fact that I forgot to mention on the last slide is, remember when we talked about the cytochrome, we said the cytochrome is an integral membrane protein within the inner membrane of the mitochondria, and it is part of the electron transport chain. So cyanide actually binds to the iron portion of the cytochrome, and when the cyanide is bound to the iron, it prevents the transfer of electrons throughout the electron transport chain, which means that you can no longer produce ATP, and that's why cyanide kills you. So I find that to be interesting. So that is the end of our discussion on the breaking down or catabolic reaction of glucose. So we're going to talk about glucose synthesis and the storage of glucose. Some vocabulary to start. Gluconeogenesis. Neo means new, so it is the synthesis of new glucose. And we make this from lactic acid, glycerol, or amino acids. Glycogenesis is glycogen formation from glucose. So first you would form glucose in gluconeogenesis, and then the glucose could be further formed into glycose or glycogen for storage. And then glycogenolysis is the release of glucose from glycogen when it's needed for cellular function. So when we have excess glucose, we can form glycogen through glycogenesis, and when we need glucose, we can break down glycogen in glycogenolysis. On the left side of this picture, it's showing glycogenolysis. So we have glycogen up in the top here. Here's our glycogen up at the top. So going down the left side, you're breaking down the glycogen to glucose, and then going through glycolysis and breaking down glucose further into pyruvic acid. The pyruvic acid is going to be broken down into acetyl-CoA, and the acetyl-CoA will enter the mitochondria, go through the citric acid cycle, and then through the electron transport chain and produce your ATP. A byproduct of glycogenolysis is lactic acid over here. On the right-hand side, we have the building up of glycogen. So we start with a pyruvic acid, and we have these several enzymatic reactions that come into place, and ending up with this last step of creating glycogen. And the main thing we want to know about the glycogen creation is that it requires UTP, which is uridine triphosphate, which is similar to ATP but with a uranine instead of the adenosine. Cells cannot use acetyl-CoA to make glucose. They cannot use fatty acids or amino acids to build glucose. They need to use pyruvate to form glucose, and then the glucose can get stored as glycogen. So that's the end of our discussion about glucose, and we're going to move on to lipid metabolism. Lipids are made of carbon atoms, hydrogen atoms, and oxygen atoms, much like carbohydrates, but they're in different proportions, which is why one is a carbohydrate and one is a lipid, but they are made of the same basic building blocks. So a triglyceride is the most abundant lipid in the body. It gets split into its component parts by hydrolysis. Hydrolysis is the breaking of hydrogen bonds, so it is breaking the bonds that are holding the different components of the molecule together. Glycerol enters the citric acid cycle after some enzymes in the cytosol convert it to pyruvic acid. So when a triglyceride gets broken down, it's one molecule of glycerol and 3 fatty acid molecules. So the glycerol is going to go enter the citric acid cycle, and then the fatty acids have a different set of enzymes that break them down to generate acetyl-CoA, and then that acetyl-CoA can move into the mitochondria. The lipid catabolism is influenced by growth hormone and thyroid hormone. Beta oxidation is how we break down our fatty acids within the mitochondria, and fatty acids are 18-chain fatty acid, can be broken down into smaller and smaller fragments. So we can, every carbon chain that enters the citric acid cycle can be immediately converted to acetyl-CoA, and then undergoing this beta oxidation process, it generates 12 ATP molecules for each 2-carbon fragment that gets removed from a fatty acid. So cells can gain, this should say 122 ATP molecules here, not 144. So 122 ATP molecules from the breaking down one 18-carbon fatty acid molecule. So fats really do produce a lot of energy when you break them down, but they are not the ideal source. And we're going to talk about that in a minute. So again, if you are super interested in understanding the molecular structure of these, this explains it, but we're going to skip over this for our sanity. So lipids provide a large amount of ATP when they're broken down, so they're an important energy reserve. They're stored efficiently in the cytosol as droplets. Remember, they are micelles. They get broken down in the digestive system into the micelles, and then they form chylomicrons. I think we're going to get into that in a minute. The downside of lipids is they're hard to access because the enzymes that are required to break them down are water soluble. They're not fat soluble, so it's hard to get at your fatty stores and break them down. Also, most lipids are processed in the mitochondria and they require oxygen. So in any time where you have, you need anaerobic metabolism, you cannot break down lipids because you don't have enough oxygen to power the mitochondria. So lipids are not a quick energy source, but they can certainly be broken down as a long-term energy source if you are undergoing starvation or having very limited glucose in your diet. Lipogenesis is the process of forming fats. So it begins with acetyl-CoA, and almost any organic substrate can be used to form fats. So that's great if you want to store fat, but it's also challenging if you don't want to store as much fat as you tend to store. So all of our cells require lipids. They maintain the plasma membranes. Our plasma membranes are phospholipid bilayers, and so every plasma membrane in your body has integral fats in them. And then steroid hormones are fat soluble, and so they have to diffuse through fat in order to reach inside their target cells and affect the target cells in the way that they are meant to. Most lipids are not water soluble, they are fat soluble, so they have to be carried from one region of the body to another, and that mostly happens through lipoproteins. So a lipoprotein is a fat that is connected to a protein in order to be transported throughout the body. Free fatty acids are not connected to proteins, and they make up a really small percentage of our circulating lipids. So here's how we are transporting lipids. So first in the intestinal tract, we talked about this last week. We have the micelles that come in through the intestinal epithelium like this, and then they turn into chylomicrons, which get filtered into the lacteals, those large lymphatic vessels that are within the intestinal mucosa. From the lacteals, they drain into the thoracic duct, which dumps into the inferior vena cava. From the vena cava, or the superior vena cava, from the vena cava, they're going to enter the heart and be sent out through the circulatory system. They can get all the way down into the capillaries, and then from the capillaries, diffuse out into skeletal muscle, cardiac muscle, adipose tissue, and the liver for utilization. Like I said earlier, free fatty acids are an important energy source during periods of starvation where you don't have enough glucose and you're forced to break down fats, or when glucose supply is limited. So if you are on a ketogenic diet and you are not eating any carbohydrates whatsoever, you have very limited glucose intake and your body kicks over into fat burning as its number one source of energy. Liver cells, cardiac muscle cells, skeletal muscle fibers all metabolize our free fatty acids. Lipoproteins are those lipid protein complexes we were discussing earlier. They contain large insoluble glycerides and cholesterol. We have 5 different classes of lipoproteins. Our chylomicrons are the largest of the lipoproteins, and we discussed those coming out of the digestive system and entering those lacteals to be sent into the general circulation. Very low density lipoproteins bring lipids out to skeletal muscle. Once they have lost some of their lipids, they turn into intermediate density lipoproteins, which are smaller. Our low density lipoproteins deliver cholesterol to peripheral tissues. These are what we call our bad cholesterol. It's one of the things when they check your blood cholesterol levels, they look for LDLs and HDLs. LDLs are generally considered your bad cholesterol, and HDLs are considered your good cholesterol. The LDLs are bringing cholesterol out to the periphery, and those are the ones that can build up in your arteries and create plaques. And if plaques become too big, they can shut off an artery and cause a stroke, or the plaques can break off and become an embolus, where then they can go and lodge in a smaller artery, like a pulmonary embolus that would lodge in your lung, or an embolus that could lodge into your brain and give you a stroke. And then our HDLs they work to bring cholesterol back from the peripheral tissues to the liver. And so they're helping you to further process down those cholesterols, and they do not seem to be involved in that plaquing process. So they are good cholesterol because they're bringing the cholesterol back. So it's a way to transport cholesterol back to the liver so it can be broken down instead of just depositing it out in the bloodstream or the blood vessels. So here's a diagram of what we were just discussing. We'll start up here with number 1. So the liver cells are synthesizing the very low density lipoproteins that get discharged into the bloodstream. In the peripheral capillaries, the lipoprotein lipase removes some of the triglycerides and so we take some of those fatty acids out and leave intermediate density lipoproteins. Those are recycled back to the liver and then become low density lipoproteins, which are bringing cholesterol from the liver out to the peripheral tissues. They are received by the peripheral cells through endocytosis. They're broken down and the cholesterol is released for use in the cell. Any additional cholesterol gets sent out into the body system where it gets picked up by a high density lipoprotein, and then the HDL is going to bring it back to the liver. The liver will extract the cholesterol out of the HDL and then utilize it for other purposes. We're going to move on to protein metabolism. So our bodies can synthesize up to 140,000 different proteins, which is really incredible. And the cool part is that all proteins are built from different combinations of 20 amino acids. So there are 20 amino acids that can be combined in 140,000 different ways to create different individual proteins. Cellular proteins get recycled in the cytosol. Their peptide bonds get broken down. They are broken into their individual amino acids, and then those amino acids can be reconstructed into a new protein. We have essential proteins. This is not in the lecture, but it is in the book. So our essential amino acids, we have essential amino acids, which are amino acids that cannot be formed by your body. There are 10 of them that we see as essential. 2 of those, arginine and histidine, can actually be synthesized within your body, but you cannot synthesize enough of them for, like in children who are growing. They need more than we can actually synthesize. The other essential amino acids must be found in the diet. So if you don't have enough, it's going to interrupt the synthesis of many proteins within your body. So essential amino acids include isoleucine, leucine, lysine, theanine, tryptophan, phenylalanine, valine, and methionine. So those all, we have to have a dietary source of all of those essential amino acids. Proteins can be synthesized. The body synthesizes half of the amino acids we need, so we can make 10, and we have those 10 essential ones that I was just talking about. The liver cells are synthesizing the carbon frameworks for our non-essential amino acids, and then amino acids get added in to form proteins as needed. So when we're synthesizing a protein, we can take this carbon framework over here on the left and use an ammonium ion. This is the process of amination. So with amination, we use an ammonium ion and an NADH, and we're adding a hydrogen and an amine or amino group over to this, the carbon structure, and creating glutamic acid. So glutamic acid is the protein that we're trying to create here. We took two different structures and we added this ammonium ion. It reacted along with this structure in the presence of NADH, which is going to provide a hydrogen. So we end up with this hydrogen and this amino group that are attached to this carbon chain that creates glutamic acid, and we have water as our byproduct here. So that's amination, the addition of an amino group to a carbon chain to create a protein using an ammonium ion. So the ammonium ion is the reactant in this instance. Transamination is exchanging an amino group between two different carbon chains. So in this one, we have glutamic acid and we have a keto acid. And then we use an enzyme called transaminase that is going to transfer the hydrogen and this amine group over to the other carbon chain. So we end up with tyrosine and a separate keto acid. The keto acids can then leave the mitochondria and enter the cytosol where they can produce new proteins. So here, in order to produce tyrosine, we are taking apart the glutamic acid, adding those molecules into the keto acid and creating a second keto acid and a molecule of tyrosine. Amino acid catabolism is the breaking down of proteins into amino acids. Proteins can be broken down, and some amino acids can enter the citric acid cycle. However, this is not ideal, and there's a couple of reasons we're going to go into. But the breakdown involves either transamination, so taking an amino group and giving it to another acid, or deamination, the removal of an amino group. And both of those require a coenzyme derivative of vitamin B6, and that coenzyme is pyridoxine. The result is the hydrogen and the amino group are liberated from the initial protein, and they come together and they form an ammonium ion. And ammonium is actually toxic to our body. And so that's a challenge with breaking down proteins, is we're creating this toxic ammonium ion. Ammonium can go through what's called the urea cycle. This is in the liver, and it makes the ammonium into a water-soluble compound that is less toxic and can then be excreted by the kidneys. But it's still not ideal to create a whole bunch of ammonium in your body. Here we've got the deamination of glutamic acid. So using a protein deaminase in the presence of water and NAD, we form an NADH, and we have removed the hydrogen and this amino group and created this ammonium ion. And we've decreased the glutamic acid down into a keto acid. In the urea cycle, the ammonium ion gets added to a carbon dioxide. They go through this processing cycle and are turned into urea from ammonium, and then the urea can be excreted by the kidneys. When our body is lacking sources of glucose or lipid reserves, our liver cells can break down internal proteins and absorb additional amino acids out of the blood and break those amino acids into small carbon chains, which can then provide ATP. The reason why proteins are a last resort for energy is because they're more difficult to break down. Carbohydrates and lipids are broken down more easily. Remember, lipids are giving a much higher yield on ATP, but complex carbs are the easiest to break down and turn into ATP. The byproduct of protein breakdown is the toxic ammonium ion. And proteins form most of the most important structural and functional components of our cells. So when we start breaking them down, we are decreasing the ability of the cells to function at their max capacity. Here's an overview of metabolism in general. This is a little bit hard to follow, but let's start with glucose. So we'll start right in the middle with glucose. So in the presence of excess glucose, glucose can be converted into glycogen. So that goes this direction. And then glycogen, when we don't have enough glucose, glycogen can be broken down into glucose. Glucose can go through glycolysis and produce pyruvic acid. It can also produce glycerol. Glycerol can then be stored as fat up here as triglycerides. Or the pyruvic acid could also go down through the Krebs cycle and create ATP. Lipids can be broken down into fatty acids, and fatty acids can go and become acetyl-CoA and enter the Krebs cycle as well. Proteins can be broken down into amino acids, and amino acids can create pyruvic acid and therefore become acetyl-CoA and enter the citric acid cycle. Or pyruvic acid, if we have enough and we don't need additional energy, pyruvic acid can then go and become turn into individual amino acids, and then those amino acids can be used to build proteins. We can also, pyruvic acid can also be used to create glucose, and then glucose can be used to store glycogen. So there's a lot going on in that picture, but it's showing us how all of these different pathways interact. And the last is how you can store fat as everything. So we looked at how glucose can become glycerol, and glycerol can be stored as triglycerides. Proteins can become amino acids can become pyruvate, then we can start to go up this chain and form glycerol and then get stored as triglycerides. So everything can turn into fat when we have too much in our system. We have 5 different tissue types that are active in our metabolism. They are the liver, adipose tissue, skeletal tissue, neural tissue, and then other generalized peripheral tissues. The liver is the focal point of regulation of our metabolism. It contains a lot of enzymes that can break down or synthesize carbohydrates, lipids, and amino acids. Our main liver cells are the hepatocytes, which we learned about last week when we did the digestive system. They have an extensive blood supply. They're always monitoring our blood and adjusting our nutrient composition as needed. And they contain specific energy reserves in the form of glycogen. So the liver contains a lot of glycogen, and it can break that glycogen down as needed. Adipose tissue is how we mainly store our lipids, primarily as triglycerides. We find adipose tissue in areolar connective tissue, within the mesenteries that are holding all of our intestines in place, the red and yellow bone marrow, the epicardium surrounding the heart, giving nice shock absorption and padding to the heart, and then also around the eyes and around the kidneys, and that's for protection. Skeletal muscles contain a lot of glycogen, so they have a huge glycogen reserve. And they're the actin and myosin, the contractile proteins. I'm sure you never wanted to hear those words again, but the actin and myosin can be broken down into individual amino acids that can be used as a resource, an energy source. So if you are starved and have no other sources of energy, you can break down your muscles, and those amino acids can then be used to produce ATP to keep you alive. Neural tissue or nervous tissue does not maintain its own reserves, and so it requires a reliable source of glucose. That's very important, that most other cells do have their own reserve, but neural tissue requires a reliable source of glucose. In the central nervous system, we can't function with low glucose, and so if you have low glucose in your central nervous system, you can easily lose consciousness. Other peripheral tissues that require energy also do not maintain large reserves, but they do have the ability to metabolize glucose, fatty acids, and some other substrates, unlike neural tissue, which only metabolizes glucose. Their preferred energy source varies from tissue to tissue, and generally they are under the control of the endocrine system. Our metabolic activity has two different patterns throughout the day. We are either in the absorptive state, which is the period immediately following a meal when you are absorbing nutrients, or in the post-absorptive state, which is when you don't have excess nutrients coming in and you're relying on your own internal energy reserves to meet your energy demands. So during the absorptive state, that usually lasts about 4 hours after a meal. So if you eat 3 meals a day, you spend about 12 hours in the absorptive state. The other 12 hours are spent in the post-absorptive state. And in the absorptive state, the primary hormone is insulin. So you're primarily regulated by insulin. So here we go, our absorptive state. We are mainly under insulin control, also some growth hormone and androgens and estrogens. In the post-absorptive state, we are mainly under the control of glucagon, epinephrine, glucocorticoids, and growth hormone. During the absorptive state, we are increasing our blood lipid levels, so we're storing lipids as triglycerides. Blood glucose is elevated, so we start to create glycogen, and some glucose can also be converted to glycerol and then stored as fat. Some pyruvate is formed into amino acids and forms additional proteins. In the post-absorptive state, the lipids are broken down into fatty acids and glycerol and then sent to the citric acid cycle. Carbohydrates, glycogen is broken down into glucose, and then glucose to pyruvate and pyruvate to the citric acid cycle. And proteins are broken down into amino acids and sent to the citric acid cycle. So carbohydrates are going to be broken down first, followed by lipids, and then proteins. When we're breaking down fats and proteins, we generate acetyl-CoA, which enters the citric acid cycle. Increased concentrations of acetyl-CoA can cause ketone bodies to form. We have 3 different types of ketone bodies: acetoacetate, acetone, and beta-hydroxybutyrate. Our liver cells do not catabolize ketone bodies, so they are absorbed peripherally and reconverted to acetyl-CoA for entrance into the citric acid cycle, but they cannot be broken down in the liver. They are fasting produces ketosis. So if you are not having a glucose form in your diet, then you end up breaking down more fats and more proteins, and you get a high concentration of ketone bodies in your body fluids. Ketonemia is the appearance of ketone bodies in your bloodstream. Ketone bodies lower your pH within your plasma, and it must be controlled by buffers to keep your pH at normal, healthy levels. Ketoacidosis is when you have dangerous levels of ketones in your blood, and they drop the pH of your blood enough that they the pH can fall below 7.05, and when the pH is that low, your body can't function in an acidic environment, and so it ends up putting you in a coma. You can have cardiac arrhythmias and ultimately die from ketoacidosis. This is unfortunately a common severe effect of uncontrolled diabetes. With diabetes, your insulin levels are, you're either insulin resistant or you don't have enough insulin, so your glucose is not getting into your cells, so your cells start to break down lipids and proteins and increase circulating levels of ketones, and that's why it's dangerous for diabetes to go untreated, because you can fall into this ketoacidosis and have that severe drop in pH and go into a coma and ultimately die from complications of diabetes. When chemical bonds between molecules are broken down, energy is released. In the cells, that energy is used to synthesize ATP by making ADP and phosphate interact with one another and creating ATP. But some energy is always going to be lost as heat. So if the body does not recapture that energy, it gets lost as heat. Calorimetry measures the total energy released when the bonds of organic molecules are broken. So in order to figure that out, this is how we know how many calories it takes to break down that donut that you had for breakfast. So the donut would be placed into a calorimeter, which is a machine that has oxygen in it, and then it has water, and the food would be burned. Calorie is and this is calorie with a lowercase c, is the energy required to raise 1 gram of water by 1 degree Celsius. Traditionally, we use calorie with an uppercase C, which is a kilocalorie. So that is the energy required to raise 1 kilogram of water by 1 degree Celsius. When lipids are broken down, they release 9.46 kilocalories per gram, carbohydrates 4.16, and proteins 4.32. If your daily energy intake, so that would be the number of calories that you're taking in, exceeds the demands that you place on your body for breaking down foods, your body is going to store excess energy in the form of triglycerides in your fatty tissue. If your expenditure exceeds your dietary intake of calories, then you're going to burn up that energy and start to burn down your energy reserves, which is going to end in losing weight loss. Our metabolic rate is affected by hormones. Thyroid hormones are the main hormones of metabolism. It controls your overall metabolism. Cholecystokinin, which hopefully from last week you remember, increases pancreatic secretions and causes contraction of the gallbladder. And ACTH, adrenocorticotropic hormone, help in suppressing your appetite. Leptin is also released by fatty tissues during the absorptive state after you've eaten. That binds to neurons in your central nervous system and helps suppress your appetite. And then the other one that we talked about last week was ghrelin, G-H-R-E-L-I-N, and ghrelin stimulates your appetite. One of the main important things in our body is temperature regulation. So your basal metabolic rate is an estimate of the amount of energy that you use on a daily basis. Any energy that is not recaptured is going to be released as heat from the body. And this is how we adjust our homeostatic mechanism of thermal regulation. Enzymes operate in a very limited temperature range within your body. So if your body is too hot or your body is too cold, your enzymes are going to stop functioning, which means most of the vital processes in your body are going to cease. As the body produces heat as a byproduct of metabolism, that happens when you increase your physical activity or your metabolic activity. You're going to generate more heat. that heat gets transferred to the water content in your body, and your body is made up of 90-something% water, so that heat can be really held onto. So in order for body temperature to remain constant, we need to have mechanisms by which we can release that heat. Remember back in A&P1 when we talked about homeostasis, I feel like that was week 1, maybe it was week 2, we talked about the internal thermostat of the body, and so that is our thermoregulatory control of our body. Heat can be exchanged with the environment in 4 different ways. Radiation accounts for half of the heat that we lose, and that is our bodies being warmer than the environment that we are in. So our bodies are at 98 degrees, 98.6 degrees. So as long as you are in an environment that is less than 98.6 degrees, which most of us are most of the time, we lose about half of our heat, or half of the heat that we lose during the day, we lose through radiation. Conduction is losing heat through physical contact with a colder surface. So if you were to sit on a cold chair, you might feel cold in the buttocks region, but that is a small region of your body, so you lose a minimal amount of heat through conduction. If you have your whole body laying on a block of ice, you would lose more heat through conduction than just sitting on a cold chair. Convection, you lose heat as air moves across your body. So it's different than just being in a cooler environment. This requires the movement of air across your body, and that accounts for about 15% of the heat loss that we have throughout the day. And evaporation is when water is evaporating off of your skin, and you lose heat while the water evaporates. That cools your skin down and decreases heat, and so we lose about 20% of our body heat through evaporation. This slide should be a flashback to A&P1 where we are reviewing. We talked about the integument and insensible and sensible perspiration. Insensible perspiration, we lose about 20 to 25 milliliters of water every hour, and that is just water that is coming out through the epithelia and evaporating off of our skin surfaces, as well as at our alveolar surfaces where we are exchanging gases within the lungs. Insensible water loss accounts for about 20% of indoor heat loss. And then sensible perspiration is when you are sweating from your sweat glands, and this happens when you are exercising, even when you're nervous, any increased activity. So the range of how much you lose in sensible perspiration really depends on what you're doing. So if you are sitting home doing nothing, you're going to lose very little in sensible perspiration. And if you are out running a marathon, you could lose up to 4 liters of water a day. Oh, sorry, per hour. 4 liters per hour if you are in severe exercising and heat mode. That's a lot of sensible perspiration. Our heat is regulated by our hypothalamus, much like so many other things in our body. We have 2 centers in the hypothalamus, the heat gain center, which is going to help you increase your heat, and the heat loss center, which is going to help you lose heat. We regulate our temperature by regulating the rate of heat production within our body and regulating the rate of heat loss from our body out to the environment. The heat loss center, so if we want to lose heat, we are going to inhibit the vasomotor center. So that's going to cause widespread peripheral vasodilation. It's going to bring more warm blood to the surface of the body, increase your skin temperature, and then you're going to lose more heat through radiation and convection from that hot skin against the cooler air outside. Sweat glands get stimulated to increase their secretory output, so more sweating, more evaporation, so you'll lose heat through evaporation. And the respiratory centers are stimulated, so you're going to increase the depth of your respiration, and so that's going to bring more air down into your lungs and allow you to lose more heat through additional breathing. The heat gain center is going to help you hold on to your heat, so it's preventing low body temperature, also called hypothermia. When the temperature at the preoptic nucleus drops, the heat loss center is going to be inhibited and the heat gain center is going to be activated. When the heat gain center is activated, we're going to have blood flow to the dermis decreasing. So you're going to keep your blood to yourself and reduce heat loss by radiation, convection, and conduction. When it's cold, your blood flow to the skin is going to be restricted and the blood flow is going to be shunted to those deeper veins. We have what's called a countercurrent exchange that occurs. So the cooler blood in those veins is going to dive deeply into our extremities, where those veins are going to intermingle with arteries, they're not going to share blood, but they're going to be lying right along one another. So the warmer blood in the arteries is going to warm up the cooler blood in the veins. So you lose some of the arterial heat. So the arterial blood going to the skin is going to be cooler, and the venous blood returning from the skin is going to be warmer. And that's going to keep more heat shunted towards your internal organs that are very important. I like these pictures. This shows in a warm environment. Over on the left, you've got blood flowing through all of your superficial veins. So we have the cephalic vein, the basilic vein, and the median cubital vein. That's a nice review from when we did circulation. And then in a cold environment, you can see that the blood gets shunted out of those superficial veins and now flows in the radial artery and the ulnar, the radial vein and the ulnar vein, and then up into the deep brachial vein. So that's up here. So when it's warm out, your blood is superficial. When it's cold out, your blood is mainly shunted toward those deeper veins. In order to generate heat, we can do that in two ways. An immediate way to generate heat is through shivering thermogenesis. Thermogenesis is the creation of heat. With shivering, we have increased muscle tone, increases the energy consumption of skeletal muscle. The more energy you consume, the more heat you produce. This involves all of our muscles, agonists and antagonists, and the degree of shivering depends on the stimulus. So depending on the outside temperature and how warm you need to be. Shivering increases heat generation by up to 400%, which is pretty incredible. Non-shivering thermogenesis is through the release of hormones that are going to increase metabolic activity. So this is more of a long-term strategy. So if you're stuck out in the cold for long periods, you're going to release hormones that are going to increase your overall metabolic activity. This can increase heat production by 10 to 15%, but it's over a period of weeks. So it really takes a long time for non-shivering thermogenesis to make a difference. So our immediate go-to is shivering thermogenesis. Thermogenesis is influenced by epinephrine and by thyroid hormones. Our infants have a hard time regulating their body temperature. They're very small, and so their regulatory mechanisms are not fully functional. So that's why we have to keep them wrapped up and really keep them warm all the time. Infants also do not have the ability to shiver. They do, however, have a lot of brown fat. And you remember back in AP1, we talked about brown fat being highly vascularized fat and containing a lot of mitochondria. So the mitochondria in brown fat can be turned on to produce additional energy. We find most of this in the shoulder blades, around the neck, and in the upper body in our infants. And we've talked before about how we're finding more in our like northern populations, the populations who are living in colder climates, and in our much thinner individuals. Our temperature oscillates throughout the day. It can fall 1 to 2 degrees Celsius at night, and it usually peaks midday or early evening, but that can certainly vary by individual. And fever, we have spent a lot of time talking about fever throughout the last 2 semesters. Fever is a body temperature greater than 99 degrees. It can occur for many reasons that are not always pathological. So sometimes we create fevers, like young children can create fevers just from exercising in warm weather. Their body temperature can increase rapidly, and then it can take a while to cool it back down. So it's best not to take your temperature if you've been out for a run or first thing in the morning. Usually you want to wait about 20 minutes after you wake up from sleep in order to take your temperature if you think you might be running a fever. So this was, I'm probably more thrilled than you to be done with this lecture. I hope you all enjoyed it. Please do utilize the discussion forum to ask me questions this week, and I will do my absolute best to explain anything and everything that did not come through as clearly as we would like in this lecture. Next week, I'll see you back for our beginning exploration of the urinary system. Have a great week.